Greetings! Welcome to our poster. In this video, I'm going to introduce an approach for identifying and mining antimicrobial resistance genes from Salmonella enterica pan-genome. I'm Yu-Wei Wu from the Taipei Medical University. To begin with, let me first introduce the idea of pan-genome. In a nutshell, the pan-genome is a way to measure the presence or absence of certain gene families, that is, genes clustered at certain threshold among different strains in the same species. It is also very often plotted into the growth curve figure, indicating how core genes, which are genes present in all strains, and accessory genes, defined as genes that appear only in some of the strains, and how they grow with the increasing number of genomes. As one can see in this figure, while the core genes remain almost stationary at the number of several thousand, the accessory genes grow rapidly with the number of genomes. This also reflects the flexibility of Salmonella enterica genomes, in which the species may be able to take in additional genetic materials and adapt to novel environments easily. And since the pan-genome indicates the presence or absence of gene families, we can very easily convert the pan-genome into a gene presence or absence table. This table may be able to be used for data mining or machine learning purposes. While it is very common to identify known antimicrobial resistant genes in bacterial genomes with the help of well-established databases, there are still novel antimicrobial resistant genes that we still have no idea about. As a result, the prediction of whether a bacterial strain resists certain antibiotics based on known antimicrobial resistant genes is still far from perfect, and we have reasons to mine additional genes from large data sets using machine learning methods. A very common approach to identify potential antimicrobial resistant genes is through machine learning feature selection algorithms. Basically, feature selection can assist us looking for the features most relevant to the prediction outcomes, which is defined as whether bacteria resist antibiotics or not in this case. So feature selection is a way to identify relevant features and ditch irrelevant ones. Usually, feature selection can identify hundreds to thousands of features out of tens of thousands of features. And very commonly, prediction model built using the selected features perform better than all features. The problem for feature selection is that the feature fits very well to the data and sometimes too well. For example, feature selection identifies features that are relevant to the outcome, and the features are utilized to predict the outcome. It is often difficult to set a cutoff to find which features are really important among selected features, and sometimes difficult to set a cutoff for the selected features if one wants to select just, say, top 100 or surpass features surpassing certain scores. So we propose a method called the cross-validated feature selection. The concept of this method is very easy, in which the original data set is split randomly into disjoint subparts. Each subparts then conduct their own feature selection. The selected features from all subparts are then intersected to find the subset of features that appear in all subsets. Also, to minimize random effect, this process is going to be conducted several times, and only features that are extracted most of the times are put into the final feature set. So we use the Salmonella enterica dataset to evaluate the cross-validated feature selection method. This figure shows prediction accuracies and the number of genes that are all features selected. One can see very easily that the prediction accuracy of the cross-validated feature selection approach is comparable to other methods, including known antimicrobial resistant genes or other feature selection methods. While the number of genes is greatly reduced compared to other methods, this shows that the gene sets selected by the cross-validated feature selection approach may indeed be more relevant to antimicrobial resistance outcomes compared to other methods. We also investigated the functions of selected genes. Surprisingly, we found that about 40 to 50% of the genes are annotated as hypothetical proteins, which indicates that their functions are unknown. In other words, these genes may be novel antimicrobial resistant genes and worthy of ongoing exploration. In addition, we checked how many genes can be annotated by the non-antimicrobial resistant gene databases. The result shows that some of the genes, up to 80% in some cases, are non-known antimicrobial resistant genes. This again indicates that the genes identified by our approach may be novel antimicrobial resistant genes. Our contribution in this work is that we propose a cross-validated feature selection approach and apply this approach to identify antimicrobial resistant genes. By testing our approach on Salmonella enterica dataset, we showed that the approach is able to select the minimum number of genes while maintaining the prediction accuracies using machine learning algorithms. We will continue investigating the gene content in order to get the most out of the identified genes.